All right, people. Can you guys hear me?
Yeah.
Cool. Good. Um, it'd be nice if some of you guys could turn on your camera so I can at least see some people. I'd appreciate it. Okay, so a little bit. Let's see. We haven't met since you guys took your test, so I wanted to tell you a little bit about that. Let me look at it here. All right. Yeah, so overall, you guys did very well. In fact, this is the highest average grade that I've had. Oh, can you please mute your microphone, whoever that was? Everyone, please mute their microphone unless you are asking a question. But yes, your guys' grade for test one was the highest grade on a cognitive neuroscience test in one of my classes I have ever had by far. I'm looking here. High B. This is probably a B plus. I have the amount of points was basically 40, and I believe it was out of 46. So yeah, that is like an 86%, which is very high. There were 31 A's, 23 B's, and there were a couple C's, D's, and F's. But overall, this is by far the highest grade I've ever had. And I'd like to attribute it to the fact that I'm sure you guys just studied your butts off for it, but it probably had something to do with the fact that it was an online exam as well, actually. But yes, nice job on that. So yeah, now I'll get to today's stuff. That's done it. Okay. So the plan for today is, I guess I forgot when I made the updated syllabus that we actually did not finish chapter 6 before test 1. So we actually need to finish some stuff from chapter 6. So we'll get started on chapter 7 next time. Next time you also have a master and his emissary quiz. I think you only have 2 more of those left, so we're almost done with the book. We do have one of those tomorrow, and then there'll be one more after that. My plan regarding your master and his emissary, your big writing assignment, I have written myself a to-do list, which includes having the formal questions posted on Blackboard this Friday. So you should have more concrete information about what you're going to have to be doing for your master and his emissary paper this Friday. I am more than happy to talk to you about your project ideas. In fact, I enjoy talking about them. That's one of my favorite parts of reading the book, is talking to students about their ideas and helping them develop them. And since we're doing everything over Zoom, it's probably not a bad idea if you wanted to talk to me, to make up a Zoom appointment, and we can kind of brainstorm ideas for your master and his emissary paper. Yes, so in chapter 6, I kind of just want to get straight into stuff. Did we talk about this study? It would have been a while ago, but have we talked about this study before? Does this look familiar? I'm pretty sure we have.
Yeah, we have.
Yeah. Of course, talking about, I know we've talked about it, but maybe it wouldn't hurt to kind of review it. We've been talking about this chapter, which is chapter 6. This is more like the perception side of things, the higher level side of things, the top-down side of things. And if you recall, chapter 5 was mostly about the bottom-up sensation, the low-level side of things. We're now on the higher side of things. And one of the perspectives on the higher level processing has to do with these dorsal and ventral pathways. Can someone remind me, what lobes of the brain does the ventral pathway involve? Does anyone remember? The ventral pathway involves what lobes of the brain, or what's the primary lobe? There's really one lobe, which is the primary lobe for the ventral pathway. Anyone remember this? Anyone? Ventral pathway involves what lobe?
Is it the temporal lobe?
It is the temporal lobe. The temporal lobe is the primary one involved with the ventral pathway. So you guys can see my screen, right? Can you guys see?
Yeah.
You can't see my desktop. Good. Okay, so This is the ventral pathway right here. It starts in the, what lobe is this right here? Anyone, what lobe is this right here?
Occipital.
That is the occipital lobe. And the ventral pathway starts in the occipital lobe and travels to the temporal lobe, and then it actually does shoot up to the frontal lobe right here. The dorsal pathway starts in the occipital lobe, but where does it go? The dorsal pathway goes to what lobe? Starts in the occipital, but goes where? No one seems to know. You guys are a little rusty. It's okay. Dorsal pathway goes up to the, what lobe is this right here?
Parietal.
That is the parietal lobe right here. So ventral pathway goes down to the temporal lobe. The dorsal pathway goes up to the parietal lobe. This was, I believe this was a PET study, or it might have been fMRI. I don't remember which neuroimaging technique this study was using. But they had participants pay attention to one of two types of information. In the objects task, people were paying attention to what the objects are on a computer screen. In the position task, they were paying attention to where the objects were on the screen. So those are the two main conditions of the study you're seeing here. What the objects are and where. Which of the two pathways, ventral or dorsal, is usually associated with what the objects are?
Ventral.
It is ventral. Thanks. I was going to go back in the slides and make sure that we talked about it. I knew we talked about it, though. Yes, ventral pathway is correct. Thank you. The ventral pathway tends to be involved with what objects are, the identity of the objects. Whereas the dorsal pathway, there's really two things that the dorsal pathway has been involved with. Determining where objects are in space, so spatial things about where things are in space, and the other about how you interact with those objects, like motor knowledge involves the dorsal pathway. So the dorsal pathway has sometimes been called the where pathway, or maybe the how pathway, because it tells you information about where objects are and how you interact with them. Whereas the ventral pathway tends to be called like the what pathway, because it gives you information about the identity of objects. And I'll just kind of get to the punchline of this study. They basically found that in the object task, which you can see the results right here, there was a lot of activity in the ventral pathway, which is shown right here. And then this little activation right here has to do with the position task. It says position retrieval minus object retrieval, which showed relatively more dorsal pathway activation here, starting down to the occipital lobe and traveling up. This is a kind of a parietal lobe activation. And so here, these are some studies that kind of support this distinction between the ventral and dorsal pathway. The evidence is really strong for the object to task. When you're paying attention to the identity of the objects, clearly it's localized mostly to the ventral pathway. Whereas when you're paying attention to the position or the where task, there's relatively more activation in the superior parts, more towards the parietal lobe. I feel like we need to do some more. I feel like we need to do some more review of this stuff, because you guys do seem a little bit rusty. So for this patient here, GS, he had a problem, and I told you guys about this. It was probably 2 or 3 weeks ago now, 3 weeks ago, where he was looking at, let's see, what was the thing he was looking at? He was looking at a combination lock, but he could not figure out that he had a type of disorder where he was not able to see it and know what he was looking at. He couldn't say it was a combination lock. What he said instead was it was a rotary phone. So by combination lock, I mean the thing that you open your lockers with back in junior high and high school. He couldn't find it. He couldn't figure out what he was seeing, but eventually he was able to figure it out using a kinesthetic code. And that's what it says here, strong use of a kinesthetic code. Does anyone remember what do I mean by kinesthetic code? How was this patient GS eventually able to recognize that what he was looking at was a combination lock? At first he thought it was a rotary telephone. Then the researchers told him, no, it's not that, try again. Eventually he was able to get it right. How was he eventually able to get it right? Anyone remember?
He turned
Is it
the yeah, yeah, go ahead.
Was that Allison speaking? Who is speaking?
Corinne.
Oh, okay. I don't see. Usually the person speaking comes up here. But yes, please continue.
Oh, he turned the combination lock, like he made the little movement.
Yeah. Yes. Yeah.
That
I see a student doing that. He'd made this movement right here. He kind of did it unconsciously. He was looking at it and he just started doing this. And then he looked over at his hand. He saw what his hand was doing. He's like, oh, it's a combination lock. But before he looked at his own hand, he didn't actually know what it was. His body kind of knew at an unconscious level how he was supposed to interact with this thing. But it was only upon actually looking at his hand performing the action that he was able to say, oh, it's a combination lock. So this type of kinesthetic code, which pathway in theory should this type of ability, the ability to know the nature of an interaction that you should have with an object, is that something that's going to rely more on the ventral pathway or the dorsal pathway?
Would it be the dorsal?
It would be the dorsal. That's one of those how type of things. Part of your knowledge about objects is how you interact with them. When you see a combination lock, one of the things you know is that part of the how for you is you make this motion when you see the thing. And so that's getting to the dorsal. That's the parietal lobe side of all of this. Whereas most of us, we would see the combination lock and our ventral pathway would be working just fine. Our ventral pathway would be fine. So we'd say, oh, it's a combination lock. But if like this patient who clearly had some type of ventral pathway damage, he had to rely more on his dorsal pathway and then looking directly at what the motor knowledge was doing, then he was able to recognize that. So that was just a little bit of review. So now I don't think we've talked about any of this stuff. And I have updated the slides a little bit. So if you have the same slides from a long time ago, please note that I have updated them slightly. So we're now kind of zooming in on some aspects of object perception. So the parts of our mind, parts of our brain that help us determine what it is we're looking at. So in theory, these are parts of the mind that involve the ventral pathway a little more. And I'll show you some evidence that that's true here in a second. But the book highlights these two abilities that human beings have, which the book makes the argument they're cool or they're it's nice that we have these abilities. And the two abilities are object constancy and cue invariance. These are the two perceptual abilities that humans and arguably some animals have. And the book makes a pretty big deal about them. And we'll just talk a little bit about some of the things that the book highlights here. Object constancy is this idea that you can recognize that an object is the same object, even if it's presented in a perspective or context or position that you have never seen it before. In other words, this is the idea, which is another textbook bold-faced term, view independence. The fact that you are able to recognize an object no matter what perspective or what viewpoint you are seeing it from. That's object constancy. And an example of that would be these things right here. This is an example of object constancy. Here are 4 different perspectives, 4 different viewpoints on this object, which is obviously like a Jeep, some type of automobile. So that's an example of object constancy. Another example of object constancy / view independence is the fact that even though part of this dog is occluded, and here there's part of the dog is kind of darkened by a shadow, you could still tell that it's the same dog. Even though the perspectives change, even though the viewpoint change, you can still understand, your brain is still able to recognize that it is the same object. So this is object constancy. In a related term would be view independence, your ability to recognize that an object is a given object, even if you're seeing it in a perspective that you've never seen it before. Cue invariance is shown in these pictures right here. You can recognize that you can recognize the identity of an object, even if its superficial features are different than anything you've seen before. So the superficial feature, which is strange here about this elephant, is, well, anyone know what's the superficial feature which is strange here?
It's pink.
Yes, it's pink. But nevertheless, even though it's pink, you still have no problem recognizing that it is an elephant. Here, looks like some type of fruit, I would guess an apple. and it's like plaid. I think you'd call that plaid, but you can still recognize that it's an apple, even though it has this weird superficial appearance. This is a human being, even though you've never seen a human being that looks just like this. This is a person. So in these, it says here, objects perceived the same even if the features change, but you could, a synonym for features is cues, which are basically synonyms for features. Even though the cues or the features are different than what we're used to, our mind has no problem ignoring the irrelevant information, ignoring the fact that the elephant is pink, ignoring the fact that the apple is plaid, ignoring the fact that the human is whatever's going on there with that little human. Our mind has no problems recognizing the fact that the cues are different than what we've seen before. Our previous experience with elephants is one color, but we're still able to see that that's elephant is an elephant, even though it's pink. So here's a study. Let's see, I need to minimize you guys for a second here. Here is a study which is supporting this idea that aspects of object constancy it's looking at some temporal lobe areas. So if we're talking about the temporal lobe, which of the pathways, ventral or dorsal, are we investigating in this study? Temporal. Say that again, please.
Ventral.
Yes, it is the ventral pathway which is being investigated here, which makes sense. We're talking about object perception now. This idea of object constancy and cue invariance. These are things which are important for our ability to recognize what an object is. So we're focusing in here on the ventral pathway. So a couple important things about the study. One, it's using a paradigm repetition suppression, which is actually pretty common in neuroscience. I'd say actually very common. There's probably hundreds, if not thousands, of studies that have been published using the repetition suppression paradigm. The idea of repetition suppression paradigm, as the name implies, is that if I present to you the same object over and over and over again, at first, when you first see the object, your brain responds very strongly. There is a large response in your brain. There's a lot of brain activity in response to seeing an object when it's the first time you've seen it. But with repetition, the brain response tends to decrease. It tends to become suppressed. This is the basic idea of repetition suppression paradigm. And the book makes the argument that the reason why you see decreased activity when you see an object over and over and over and over again is because the brain and your mind gets more efficient at processing the object, and so it doesn't have to work as hard to recognize what it is that you're looking at. That's the basis of repetition suppression. So now we're going to talk about a study, and I guess I should go to the right here. Yeah. Here are the results of the study. So it says left fusiform, right fusiform. You might want to write in the margin or your notes here. That's just part of the temporal lobe. The fusiform area is usually considered the inferior or the bottom part of the temporal lobe. So we're talking about the ventral pathway here, part of the temporal lobe. And the stimuli in this is a repetition suppression paradigm. And there's some interesting findings here. They were looking at the left temporal lobe, that is the left fusiform area, and the right temporal lobe, the right fusiform area. And you can see the types of objects that they were presenting to people. They were using neuroimaging. I think this is, bold, so it looks like it's fMRI. They're looking at using fMRI. They are measuring the brain activity in the left and right temporal lobes for the different types of objects you see here. So a new object is basically the first time you've seen an object. You've never seen it before. And consistently with what we've been talking about, there's a lot of activation here and a lot of activation here. Out of all of the conditions, some of the highest activation occurs the very first time you see an object, because the brain, in theory, has to work a lot to process the identity of the object. And so there's more bold response, more blood flow into that brain region because the brain region is having to work more hard. So that's the first condition. They see a new object. But then these blue bars represent when the object is old. So when the object has been repeated a bunch of times. However, there's a little twist to this, literally, where, here in this bar right here, and in this bar right here, you're seeing the same object that you've seen earlier, and it's presented in the exact same perspective. So like, it's literally, you saw an object that looked like one way earlier, and you're seeing the object looking the exact same way, the exact same perspective, the exact same viewpoint now. And not surprisingly, we're seeing repetition suppression. When you see an object again, that you've already seen before, the brain's response tends to decrease. But the interesting thing about this study is that there's a hemispheric asymmetry. That there's a difference between the left and right hemispheres in the way they respond to one particular type of condition here. And you can see that it's in the different view right here. So what the different view is you're seeing the same object that you saw earlier. So it's repeated, but this time it's been rotated. It's been somehow changed just a little bit where it's been like, I think it's easiest just to think about it. It's been rotated. So instead of seeing the object in the exact same viewpoint, you're seeing the same object, but now it's been twisted. And the question is will the brain respond as though this is the exact same object that it's seen before, or will the brain respond as though, oh my gosh, this is a completely new object that I've never seen before? You take the same object, but now you're rotating it. Does that rotating make the brain view this as a new object? Or can the brain still tell, oh yeah, this is the same object, it's just rotated, that's not that interesting? And the interesting thing is that we find that there's a difference between the left and right temporal lobes and how they respond to this different condition. So I want someone to tell me, which hemisphere of the brain views the rotated object, that is the object in a different view, as though it's a completely new thing that it's never seen before?
The right.
That is correct. Can you explain how you knew that?
So the response for the different view is the same, like signal. The bar is the same as the new object.
Yes, exactly. So you focus in on this different view in the right hemisphere. And basically, if you compare it to the new objects that they saw earlier, the brain is responding the same way. So it's like in the right hemisphere, you take an object and then you rotate it. The right hemisphere is like, what? I've never seen one of these things before. But if you do the same thing in the left hemisphere, you take that object and then you rotate it. The left hemisphere is like, I've seen this before. This isn't interesting. And so there's a couple things I want you to know about this study. One is I want you to understand the repetition suppression paradigm in general. That is, with repeated exposure, your brain's response decreases. But it's also cool to see how the left and right hemispheres respond differently. The left hemisphere is able to tell that it's the same object, even if it's rotated. But in the right hemisphere, when the object is rotated, it's as though it perceives it as a completely new object that it's never seen before. Any questions or comments about this study? All right. Moving on. So this was getting at object constancy, view independence. The idea that your brain, to some extent, is able to recognize that an object is the same, even if it's presented at different perspectives. Particularly the left hemisphere is good at that. You can change the object in different viewpoints, and the left hemisphere can tell it's the same object. The right hemisphere, not so good at that. That was object constancy or view independence. Now the other term that I was talking about a few slides ago was cue invariance. And so this is not part of your textbook. This is taken from an actual publication on, I think in the study they were measuring neuronal responses in animals using like they put electrodes directly into the animal's brains and were measuring in part of the temporal lobe. And it's cue invariance. And the reason I'm presenting this picture is because it shows another type of cue invariance. Like the same object is presented here, but the cues are different. Could you guys tell me what is the object which is present in all three of these different pictures?
A square?
Yeah, it's a very, very simple object, but it is just a square. And it's good to kind of see, that cue invariance is the idea that your brain, it's very easy, it feels very easy at least, is able to recognize that it's the same type of object, even when the superficial features are different. This one, I believe, stands for luminance. This one is texture. Kin stands for movement. Those arrows represent the fact that there's a movement. And what they were doing in this study, here's the abstract. They were measuring, does it say the brain region? I know it's the ventral pathway is what they're measuring in. I'm not sure. Yeah, inferior temporal cortex in macaque, which is a type of monkey. They were measuring neurons in the inferior temporal cortex. And basically what they found was that there are neurons that were cue invariant. That is, there are neurons in this part of the temporal lobe that respond to square, even if the superficial features are different. There are also specific neurons in the temporal lobe. Interesting, at least there's been some studies which actually suggest that there are neurons in your temporal lobe that will respond to specific people. So there's some studies about how there are specific neurons that respond to like famous faces in a given individual. And you present that famous face and the same sets of neurons tend to respond to that face, and a different set of neurons will respond to a different face, even if the cues of that face are different. So you see a full color picture of me, and you're going to have a set of neurons in your mind, in your brain that respond. But then you see like a black and white picture of me. The same neurons are still going to respond because those neurons are cue invariant. There are neurons in your temporal lobe that recognize specific objects, even if the superficial features of those objects are different. Okay, so moving on now. This is a really fun part of the chapter, I think, where we're talking about some of the what happens when individuals, when some of their top-down, their higher level abilities to recognize objects are impaired. So these are individuals who we're going to be talking about who their vision and their hearing are fine, but nevertheless, they have problems recognizing objects. So the problem is not in their sensation. It's not in the fact that they're bad at seeing or they're blind in any way. The problem is at the higher levels of their mind, the top-down parts, the perceptual aspects of their mind and brain are impaired, which prevents them from being able to recognize certain types of objects. And I really like this, the distinction that the book makes between agnosia, which is basically this impairment. Agnosia is basically an impairment in object recognition, versus general memory loss. And it's important to understand this distinction because agnosia would be, if you have visual agnosia, for instance, you are impaired in recognizing visually presented objects, but you would still be fine at recognizing auditory objects. So an example, by the way, of an auditory object would be like a melody. So a person with visual agnosia would have problems recognizing visual objects, but they're still good at auditory recognition. Another type of object recognition that we don't think about is like right now my eyes are closed. And if you put something in my hands, I could use my tactile sense and I could feel, oh, this is a coffee mug that I'm holding right now. And I'm recognizing this object with my sense of touch only. People with agnosia, usually their impairment in recognizing objects is specific to some modality. So you have visual agnosia. Your sense of vision is actually okay. You might have 20/20 vision, but people with visual agnosia are going to have problems recognize objects when they see them. However, these same individuals, if you touch the object, they should be able to recognize it. So their problem is using their sense of vision to connect that to some type of memory that allows them to recognize, oh, when I see the coffee mug, that's a coffee mug. But as soon as they touch it, they would be able to recognize it. But relying only on their vision, they would have a problem. And so the book does a really helpful job at distinguishing between a person with agnosia versus a person with memory loss. So here is an individual with agnosia. Right here, the person is using their vision only. They're looking down at the fork. And this individual is unable to recognize, you can see the question mark right there, unable to recognize that the thing that they are looking at is a fork. However, the same individual, if they pick up the fork, or if I pick up my mug, all of a sudden they're able to say, oh, it's a fork. And the reason that they're able to do this is because agnosia is usually specific to one sensory modality. This person clearly has, will you tell me, does this person have visual agnosia, auditory agnosia, or tactile agnosia?
Visual.
It would be visual agnosia. This person is unable to recognize this type of object using their sense of vision only. If you let them touch something, use their tactile, they're fine. So the impairment is specific to their visual modality. Here is a person, though, with arguably a more extreme impairment. This person, the book makes the argument, has just general memory loss. So this person looks at the fork and using the visual modality doesn't know it's a fork. Then you give this person, you put the fork into their hand, and they still can't tell you that it's a fork. And so this person is unable to recognize the object, even when you're giving this person information from multiple sensory modalities, which suggests that for this individual on the bottom right, that their impairment is general. They just have a problem with object recognition in general, which would be considered just a general memory impairment. It's like this person does not have a memory of the object fork anywhere in his brain or mind. This person clearly does have the memory of fork. They're just not able to retrieve it with visual stimulation. So I think the book does a really good job here of distinguishing between agnosia, which is sensory specific, and memory loss, which is sensory general.
Can you be born with agnosia, or is it something that like you can like it's brain damage and you develop?
I believe usually it occurs from brain damage, but I would not be surprised if there actually are people that are born with some type of biological, genetic thing, which makes it, that predisposes them to agnosia. But I believe the majority of the times it's caused by brain damage.
I have a question about prosopagnosia.
Yeah.
So if agnosia can work with both visual and tactile, could somebody remember their face but forget what they sound like? Like, does prosopagnosia work all the ways that agnosia works too?
That's such a great question. And my guess would be that it is purely visual, but I don't actually know if anyone's done that study. It's such a good study idea. If no one has done that, you should totally do that, because that's a really good study idea. The problem is that prosopagnosia is exceptionally rare. By the way, prosopagnosia is face blindness for people that the inability to recognize faces. And I believe my hypothesis would be is it's visual specific. So if you can't recognize a person's face, if you heard their voice, you would be able to recognize the person's voice. That would be my hypothesis. But I've never actually seen that study done. Really interesting question. Thank you for asking. Okay, so a couple things about agnosia. We're going to kind of dig into agnosia a little bit. First, I want to distinguish agnosia from optic ataxia, which I think I have optic ataxia. Optic ataxia is a deficit in visual manual guidance, which is basically a fancy way, in my opinion, of saying like hand-eye coordination. Optic ataxia is kind of your ability to see something in space and be able to reach out to where that object is, no problem. So using your sense of vision to guide where you're going to move parts of your body to interact with objects out in the world. Versus agnosia, which once again is a sensory-specific impairment in recognizing an object. So, and theoretically, although the book talks about how things are not as simple as this distinction is made between the ventral and dorsal pathways, theoretically, agnosia, which is an object recognition problem, theoretically, that problem should be tied more to impairments of the ventral pathway, that is the temporal lobe, whereas ataxia should be associated more with impairment or lesions of the dorsal pathway, that is the parietal lobe. So those are hypotheses. The evidence is kind of mixed on this, but in theory, that would be like the more straightforward hypothesis that you would make regarding the ventral and dorsal pathways and their contribution to these two conditions. We're more interested in agnosia in this chapter. And here is one type of agnosia right here. So aperceptive agnosia, which the book, I'll kind of read this here. Problems integrating spatial information, especially if the objects that you're looking at are presented in a odd viewpoint. If it's a viewpoint or perspective, an orientation or position that you've never seen before, or that it's rare, it's not a common perspective, then people with aperceptive agnosia particularly have problems recognizing the object. So here, you're obviously looking at hats, and then this would be like the not typical position. And I think the thing that makes this less typical is there's dark shadows over the hats. Here is a cat. And then can you guys tell what is unusual about this picture of the cat?
You see the back of it?
Yeah, you see the cat from behind. And so presumably this is like the stereotypical, the quintessential, like your mental picture of a cat, you kind of imagine a cat looking at you. You don't really think about what do cats look like from the back. And that's what this is right here. People, according to the book, with apperceptive agnosia are particularly impaired in when the abnormal positions, the abnormal like this, and on this one right here. And here's some interesting brain data that were also presented in the textbook. Anterior versus posterior damage of the left and right hemispheres. And really these two graphs are showing the exact same thing. So really you only need to look at one of them. This is just the graph for the hats. And this is the graph for the cats. And they're basically showing the exact same pattern of data. And the pattern is, if anterior damage in this study, I'm pretty sure they mean like towards the frontal lobe. They don't actually specify what lobe. Anterior usually means towards the frontal lobe. You have damage to your frontal lobe, you're really not that impaired in this task. If you have damage to the posterior parts, which I believe probably means parietal, but also occipital lobe and maybe some parts of the temporal lobe. So yeah, like temporal, just all the stuff back here, but it'd probably be parietal, temporal in particular, maybe a little bit of occipital lobe is generally what posterior means. They show that there's relatively more impairment for recognizing these not typical objects. And you tell me, which hemisphere, if you have posterior damage, if you have damage to which hemisphere tends to cause more serious impairments in your object recognition? Can you guys tell by looking at this?
Right hemisphere?
Yeah, the right hemisphere for sure. If you have damage to the right hemisphere, your ability to recognize these objects is particularly diminished. It's particularly impaired, at least according to this study. So in general, posterior lesions did cause more problems than anterior lesions in recognizing not typical objects. But in particular, if you have posterior damage to the right hemisphere, at least in this study, that really caused problems. That really increased the likelihood that you would be diagnosed with apperceptive agnosia. Any questions or comments about this study? Okay, here's another one. This is integrative agnosia, right? Yeah, integrative agnosia. And so, the book talks about a case study of an individual here. I like just going straight to this task right here. So the task here is that you see this thing, this little Mickey Mouse looking thing. It kind of looks like Mickey Mouse a little bit to me. And your job as the participant is to just draw this. And in fact, I can actually do this. I can actually take the time and draw it since I have the thing here. Okay, so the way most people would draw this who are not impaired, people that do not have integrative agnosia, would be something like this. I'll draw this square, and then I'll draw this circle, and then I'll draw this square right here. That's how most people would draw this. That is, they would draw each shape one at a time. But if you look at, and I'll go back to the slides here now. If you look at this right here, this is an individual with integrative agnosia. I think this is CK, the individual from the book. Instead of drawing each shape, like each square separately and the circle separately, they kind of draw it as one contiguous object. And you can see this by the numerical scheme here. So they start up here 1, then they draw this 2. And so they drew 2 lines of the square, and then they draw a part of the circle 3. Then they start drawing the other square, and then they do draw that as one thing. But 7, and then they pick up their pencil, go over here, finish the square over here, and then they finally finish the circle over here. So instead of doing what I did, which I just drew the square, and then the other square, and then the circle, an individual with integrative agnosia is more to kind of have this odd kind of strategy. And the textbook kind of makes a suggestion. Patients with integrative agnosia do not see objects holistically. That is, they have problems recognizing that, this is an entire object right here, and that the entire thing is, an entire object. Instead, they kind of break it down into the fundamental pieces, and they kind of just trace each piece by itself without really getting a sense of the big picture that they're looking at. That would be integrative agnosia. I think this was pretty straightforward, and usually on the test, students actually do pretty well on this question. But does anyone have questions or comments?
I have a question. So does it only work that way if they're asked to draw something, or if they're asked to describe it? Do they describe it drawing one line or next, or do they describe it as two squares in a circle?
That's a really good question, and I don't remember if the book mentions that or not. My guess is that they would not realize that it's two squares in a circle. That's such a good question. You guys are on fire today with your questions. I just don't know the answer to that. It's another empirical question. I would hypothesize that they would not realize, they would not be able to describe it as two squares in a circle. Because if they were able to describe it that way, you would think that that's the way they would trace it. So my hypothesis is they would not be able to describe it. That's a really good empirical question. And if you were able to perform that study, it would be publishable, unless someone's done it before. But I don't know if anyone's done it before. Really good question. Thanks for asking. Any other questions or comments? Okay, so here's another cool, another kind of agnosia. And there's, this is associative agnosia. This is the idea that when you see an object, your mind is, individuals with associative agnosia definitely do not have integrative agnosia. So individuals here, they are able to perceive things holistically. And you can tell because here, this is why this exists. This is like kind of all these objects kind of thrown into the jar, and then they took in a person with associative agnosia. And you can tell that this person does not have integrative agnosia. Can you guys tell, looking at just any one of these, what is this participant doing that you can clearly tell that they do not have integrative agnosia?
Is it because they're able to draw the objects just fine?
Yeah, they're able to color them in just fine. The objects were already drawn for them. They just had to color them in. But they're able to color them in just fine. So an individual with associative agnosia knows that this is a single object. And even though it's touching this one, they know that they're two different objects. They know that this is one object and this is another, which allows them to separate them spatially. They're able to see this is one object, I can perceive it holistically. This is another object, this is another, and they can separate the objects in their mind and separate them in space. What individuals with associative agnosia are not able to do is they're not able to, and as it says here, assign meaning to the objects. So I have two great examples. I really like both of these examples. So one example, so you have to kind of imagine this in your mind. There was a study in which the researchers, they took like, maybe I should actually draw this on the iPad. They took a picture of an elephant, and I'm tempted to try and draw the elephant. Oh man, that's pretty bad. Looks more like a squid, but that's an elephant. All right, write in your notes. They're all right here. Elephant, right? That's an elephant. Pretty bad elephant, but you get the point. They took the elephant right there, and then they took another picture of a mouse. Right, I believe it was a mouse, if I'm remembering correctly. But they blew it up. They blew up the picture of the mouse. So physically, oh my God, drawing a mouse. Are you kidding me? Let's see. Draw the whiskers right there. Oh, and I erased it. Damn. I don't know how I did that. But you get the point. They draw the picture of an elephant, and they put it right next to a picture of a mouse, which would be right here. And they made it so that in the pictures, they blew up the mouse, they shrunk down the elephant. So in the pictures themselves, the two objects were the exact same size. So now let's go back. And then they asked this patient, FRA, which of these objects in real life, they would say, so the objects you're seeing here are, they're the same size in these pictures, these two objects that you're looking at, these two pictures. In real life, which animal that you're looking at here is bigger? And obviously the answer would be, well, just make sure you guys understand, what is the animal which is bigger in real life?
Elephant?
Yeah, the elephant is the bigger animal in real life, of course. But the people with associative agnosia, they weren't able to answer this question because they were not able, the pictures they were looking at were changed so that the elephant and the mouse were the exact same size in the pictures. And in order to answer the question, which one is bigger, you have to have you have to be able to say, okay, this is an elephant. And then you connect that picture to the meaning of elephant. The elephant is a big animal that has a trunk. And they can feed themselves with their trunk and stuff like that. And they can spray water with their trunk. A mouse is a small animal. It's a rodent. It's nocturnal. And so what you're doing here, what we can do very easily is we can connect the visual appearance of the objects to the meaning of those objects. And part of the meaning of the objects for us that we can seamlessly retrieve is elephants are big, mice are small. Therefore, we know that the picture of the elephant is bigger. So we would easily point to the elephant as being the bigger object. But individuals with associative agnosia are going to have trouble with this because they see a picture of the elephant and the mouse, and they do not quickly and easily connect those pictures to meanings, which is hard for us to even imagine. It's hard for me to imagine seeing an elephant and not realizing, oh, elephants are a big animal, seeing a mouse and not realizing that mice are small animals. But presumably people with associative agnosia do have this problem. Here's another example. I don't think this one is in the textbook. But this is another example which I think really helps to understand associative agnosia. The task here, which is kind of a common task throughout cognitive psychology, is you're presented with this. This is the object is like the standard. And then you're shown two objects beneath it. And your task as the participant is to indicate which of the two objects below is most similar to the object on top. And it's kind of a trick question because both answers are right. So first off, why is this answer right? What is it about this shape which is similar to the top one?
It most closely looks like the image on the top.
Yeah, it's the shape. The shape of these two objects is very similar. The shape is the same. So if you say, well, I think if this one is most similar to the top one, you're basing that response on the fact that you prioritize the shape, the visual appearance of the object itself. However, you could also say that this object is very similar to the top one. Why would that be the correct answer?
They're both umbrellas.
Because they're both umbrellas. So now the question for you I have is, which one of those two types of responses, either this one or this one, requires that you connect the picture of the object to your knowledge of the meaning of that object? Which answer, this is the one on the left or the one on the right? Which one of those requires you to be able to connect the picture to your knowledge of the meaning of that object?
The image on the right.
Yes, the open umbrella, the image on the right, that requires you to process something about the meaning. This one right here, you're just looking at the superficial features and you're saying, oh, the shape of this is similar here. But in order to make this answer, you have to say, oh, this looks like an umbrella. And an umbrella is a thing for, protecting yourself from the rain. And sometimes you take an umbrella and you open it up. And when you open it up, it looks like this. And then you say this, oh, this is an umbrella in the open position. Therefore, these two things are the same. So individuals with associative agnosia, will you tell me, individuals that cannot connect the meaning, the visual object to the meaning, well, it's probably pretty obvious by now. They're more likely to choose this one right here. Control people are probably slightly more likely to choose this one than this one. Individuals with associative agnosia are way more likely to choose this one because they are not able to connect the visual appearance of the object to the meaning of that object. And so they focus more on superficial features of the objects, like their shape, what they look like. Any questions or comments about associative agnosia? All right. Okay, so now we're at face perception, and someone was asking about prosopagnosia. And this is actually something we've talked about before. We talked about this in the chapter on hemispheric differences, way back, and that was chapter 4. We're going to talk about it again. It's another type of object that you can recognize. Actually, humans seem to be very good at recognizing human faces. And we're interested in the brain mechanisms that allow us to recognize faces. And the evidence presented in the textbook does suggest that there's a whole network of brain regions, including the occipital, temporal, and parietal lobes, that are involved in helping us recognize faces. Now, the ones that there's been the most research on are the occipital and temporal, which makes sense because these are part of the ventral pathway, which we know is important for object recognition. Faces are a type of object. And the occipital and temporal lobes in particular seem to be very important for face perception, although it does appear that the parietal and maybe to some extent the frontal lobe are important as well. So basically all lobes of the brain are important, although the occipital and temporal are particularly important for face recognition. And so when I have some brain, we have the brain lesion data that I'll show you again. Prosopagnosia is face blindness, difficulty recognizing a face that you have seen before. And if we're talking about which hemisphere is more important for face blindness, it's pretty clearly the right hemisphere. However, I want to remind you of the facts that we talked about way back in chapter 4, which is the brain is kind of like an airplane, and it has two engines. It's got one on the right and one on the left. And just like an airplane, if one of the engines goes out, for most people, you can still use the other engine or the other lobe of the brain to perform a task. So the right hemisphere is definitely more important for face perception than the left hemisphere. But for most individuals, in order to develop prosopagnosia, both hemispheres of your brain have to be lesioned. It's like both engines of the plane have to catch on fire or whatever in order for you to develop prosopagnosia, because you are able to recognize faces pretty well in most people with just one hemisphere or the other. So let's look at the data. These are taken from your textbook. Let's see, and here it talks about the temporal, parietal, and occipital lobe. And here's people with bilateral damage to only the left hemisphere, damage to only the right hemisphere. So it appears that the occipital lobe is the most important lobe because, for instance, 91% of the people that developed prosopagnosia after bilateral lesions, they had lesions in their occipital lobe. 61% of those individuals had lesions in their temporal lobe. And only 9% of those individuals had lesions in their parietal lobe. So this is consistent with what we've talking about. Will someone remind me, which pathway is most important for object recognition, telling you what it is you're looking at?
The ventral.
It is the ventral, and this is consistent. The occipital and temporal lobes are both part of the ventral pathway, and damage to the ventral pathway is more likely to develop into prosopagnosia. But I also want to remind you, we talked about this way back in chapter 4, but just in case you forgot, this is the sample. These are the amount of people that had prosopagnosia. 46 of the people in this study out of 71, 46 people had bilateral lesions. So everyone had prosopagnosia. 46 of those people got prosopagnosia with bilateral lesions. Only 4 of those individuals developed prosopagnosia after a unilateral lesion of the left hemisphere. And 21 of those individuals developed prosopagnosia after unilateral lesion of the right hemisphere. So overall, the ventral pathway is really important for face perception. The right hemisphere appears to be more important than the left for face perception. But in most individuals, in order for you to develop prosopagnosia, that is complete face blindness, you have to have both hemispheres damaged. You have to have bilateral lesions, because even if one hemisphere is damaged, in most individuals, you'll still be able to use the residual functioning in the other hemisphere to help you recognize faces. Any further questions on prosopagnosia, face recognition? I guess I have some more that I want to talk about. So yeah, I'll just keep going, but I'll let you ask questions in a second. So this is fMRI study, it looks like, where they're looking at what regions of the brain are more activated when you're looking at faces compared to when you're looking at objects. So one condition, you're just looking at faces on a computer screen, and another condition, you're looking at objects like this on a computer screen. And they use fMRI, functional magnetic resonance imaging, to determine which parts of the brain respond more to faces than to these objects. And this is what's shown here. Now, I want to draw your attention to something very important, is that this image is flipped. And so you want to make a note of this. I think it might say it in the notes portion of the slides as well. Although this appears to be the left side of your picture, this is actually the right hemisphere of this person's brain. And although this is the right side of your picture, this is actually this person's left hemisphere of the brain. So it's like the opposite of what it looks, unfortunately. And I think it does say that again in the notes portion of the slides. Right here, though, this is brain activity in the inferior temporal lobe. This is brain activity in the right inferior temporal lobe. This is brain activity in the left inferior temporal lobe. And you can see that consistent with the brain lesion data that we just saw, both hemispheres, both temporal lobes become activated when you're looking at a face. But will you tell me which hemisphere appears based on which hemisphere out of these two appears to be more important for face perception? This is the right hemisphere, which is the left. In other words, which hemisphere appears to have more activation when you're looking at faces?
The right hemisphere.
Yeah, and you can see this is just a bigger chunk right here. And it's also a hotter color. That white means it's a lot of activation right there. So there's more activation here in the right hemisphere when people are looking at faces compared to over here. It is bilateral. So you get activation in both hemispheres. It's not all on one side or not on the other. It's using both hemispheres, but it's clearly more activation in the right hemisphere of the people's brain when they're looking at faces. And then I guess we'll talk about, this is another type of object that people recognize, and that is words. This is another type of object, and it's another type of object which quite clearly is connected to the ventral pathway. One of the more striking impairments that occurs is when people have damage to the left inferior temporal lobe. So that is the left ventral pathway, but particularly the inferior part of the temporal pathway, are very likely to have basically word blindness, a visual word blindness, where they have problems reading words. We all read words now, at the college level, and for a long time you've been very good. You read words, and you can kind of very easily, automatically, you read the word without even having to think about it. Individuals that have damage to this region of the brain called visual word form area, which is the left inferior temporal lobe. Visual word form area is the left inferior temporal lobe. If you have damage to this region of the brain, then you are very likely to develop this type of word blindness where you cannot read words quickly. You can still read words if your visual word form area is impaired or destroyed, but it's just it's a very difficult process. You have to sound out the words kind of like a kindergartner does or a first grader when they're reading words. So this seems to the left visual word form area, this left inferior temporal lobe, seems to be a part of your brain which is important for seeing a word and just automatically being able to read the word without even having to think about it. When it's impaired, you have to resort back to a sounding it out strategy like you used when you were acquiring your reading abilities when you were young. One thing I want to point out here is that this is the left hemisphere. So unlike the other picture that I showed in the previous slide, this now is the left hemisphere. This is the left hemisphere too. These left and right things, in case you're curious, this is a hemifield study. This means the word is presented in the left hemifield. This means the word was presented in the right hemifield. And when the word is presented in the left hemifield, you do see some activation in this part of the right hemisphere. But one thing which is important is whether the word is presented in the left hemifield or the right hemifield, you see activation in this one part of the left hemisphere, the visual word form area. It's a pretty strongly lateralized part of the brain, which is really important for expert abilities in reading words. Once you've been trained to read words, it's a region of the brain which becomes highly activated. I'm not going to require you to know this stuff about V4. Well, I guess I'll say it though, just to avoid confusion. In V4, when the word is presented in the left hemisphere, you see there's activation in the right V4. When the word is presented in the right hemifield, you see there's a lot of activation in left V4. I guess it's important. What lobe of the brain is V4 in? Does anyone remember? I'll give you a clue. It's in the same lobe of the brain as V1.
Occipital?
It is the occipital lobe. So here we're looking at occipital regions, and the occipital regions are clearly contralaterally organized. If the word is presented in my left visual field, then my right occipital lobe becomes activated. If the word is presented in my right visual field, the left occipital lobe is activated. However, when it comes to the part of our brain which is activated in the temporal lobe, this is the inferior temporal lobe right here, this is the visual word form area, no matter which visual field the word is presented in, you see activation in the left visual word form area. This is a kind of complicated topic, so I wouldn't test about it, but it is one of the more interesting developments over the last 20 years, is that it's becoming clear that these hemifield contralaterality effects that we see are more likely to occur at low-level parts of the brain. That is, when something's in the left hemifield, it projects to my right hemisphere. That occurs, the contralateral organization occurs early in processing, and later processing, like the one that occurs in visual word form area, it doesn't matter whether the object is presented to the left or right visual field. You're going to see that lateralized processing in the left hemisphere more. The thing that I want you to know most about this slide, though, is that the visual word form area is in the left hemisphere. It's in the inferior temporal lobe, and it's involved with reading words quickly and effortlessly. The last topic is mind reading. And I'm not going to say too much about this. I just want to say that mind reading it's a way of, you measure brain activity when people are looking at different types of objects. And so, like, for instance, here is, looks like a house or a building. Here is a picture of a face. And what you do is you use a computer, and every time the person is looking at a face, the computer basically says, okay, these are the parts of the brain that are activated when the person is looking at a building. And then the same computer, when you're looking at the face, the computer's like, okay, and these are the other set of brain regions which are activated when the person is looking at the face. And the computer starts to learn, okay, this is what the brain's doing when the person's looking at a building. This is what the brain's doing when they're looking at a face. And eventually, you can train the computer that even if the computer does not know what the person is looking at, so the computer doesn't know that you're looking at a building or you're looking at a face, eventually the computer can tell by looking at your brain activity. So I am looking at a building. I'm looking at a building across the street right now. And if there's a computer, and if my brain was hooked up to an fMRI machine, and the computer could kind of read out what the way the bold response was occurring, the computer could read out the bold response. It could read out the brain activity and be like, oh, Rosenthal right now is looking at a building. I can tell because these parts of the brain are activated right now. Those are the parts of the brain that in the past had always become activated when Rosenthal was looking at a building. Therefore, Rosenthal is looking at a building. And then I could look at a face, so I'm looking at a picture right now. The computer could do the same thing. The computer would say, oh, now a different set of brain regions are activated in Rosenthal. These are the brain regions that early on during the training phase, every time Rosenthal looked at a face, these are the brain regions that became activated. Therefore, Rosenthal must be looking at a face right now. And so this is mind reading. It's the ability for computers to start looking at our brain activity to figure it out exactly what it is that we're looking at. And I think I want to call it a day there. I'm going to start here. So we have literally one slide for chapter 6. I do want to give you a little homework assignment though, which is watch this YouTube video. It's pretty short. Watch this YouTube video about individuals with locked-in syndrome so that when I talk about this next time, you'll understand a little bit. It should be a pretty quick video here. Go ahead and watch this video right here, and then we'll start out talking about this stuff next time in chapter 6. It's just the one slide. Then we'll get right into chapter 7 next time. So I'm done. If anyone wants to stick around and talk, we can. Otherwise, I will see you Wednesday. Sorry, no, I'll see you Friday, rather. I'll see you Friday.
I have a question.
Yeah.
So people with prosopagnosia, how would that affect